Your career begins.
எனக்கு எதுவும் வீடியோ விசிபிளா இல்லையே.
இப்ப நடுவுல கட் ஆயிடுச்சு.
Good afternoon, everyone. On behalf of Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology, I warmly welcome you all to the another session of Ocean Research Knowledge Series 2026. So, it is a continuous expert lecture series that brings together scientists and researchers to share the latest advancements and innovations in ocean and marine sciences. Moving on to the session, today expert lecture is on macrobenthic communities as bioindicators of coastal environmental health. And we are happy to have Dr. P. Achyutan, Assistant Professor Research, Center for Ocean Research, as our speaker. Over to you, sir. Now you can share your screen.
Thank you, Anu. My slides are visible.
Yes, sir, it is visible, sir. You can proceed.
Okay, thank you.
Thank
Good afternoon, one and all. And today I'm going to discuss the role of macrobenthic community as a bioindicators of coastal health. So those who are new here, myself Achyutan, I'm assistant professor at Satyabama Institute of Science and Technology. Um, Let me introduce first what is macrobenthic organisms. The macrobenthic organisms, the organism that live on sediment or within the sediment, and organisms including polychaete, crustaceans, mollusk, and echinoderms. Those are called it as macrobenthic organisms. And those organisms, they are closely associated with the sediment. So anything happens in the sediment, like pollutants, which is deposited on the sediment, which directly impact on the macrobenthic organisms. If any changes in their abundance, diversity, and community composition, that will give you the valuable information about the um, coastal ecosystem. My next slide. Major coastal environmental stresses. Coastal ecosystems are exposed to the multiple pollutants like domestic and industrial waste, heavy metal, emerging pollutants, and so on. The list keep on going. Since these organisms, they are tightly associated in the sediment, and whatever impact which is pressure on these macrobenthic organisms, those organisms will directly indicate them. As the example of if organic load is high, the polychaete, specific species of the polychaete abundance will be high compared to the other organism. Like that, examples many other. Why macrobenthic organisms? Why macrobenthic organisms are good bioindicators? Macrobenthic organisms are excellent bioindicators because some are very tolerant to the pollutants. and opportunistic species, the species which benefit from the pollutants. Those are called as opportunistic species. And some organisms are very sensitive. If any impact happens, their abundance and their diversity drastically will go down. So based on these stresses, those organisms simultaneously indicate those organisms, we can call it as biological indicators. Next slide goes to community response to environmental stress. If the healthy habitat generally supports the diverse organism, for example, is there any community or any any community is there? If there are healthy organisms, they, as an indicator, they shows diverse organisms like very abundant organisms, very high numbers of the organisms. If any pollutant or moderate rate of pollutants, the sensitive species will drastically will go down. That indicates there is some pollution impact on them. And there's some organism which is tolerant, their abundance go up. So based on that, we can conclude that this area is polluted for specific pollution. What are the organisms indicates what are the pollutants? This slide goes to that. The polychaete as one of the high abundant organisms in any coastal ecosystems. There's, as I told you, some are very sensitive and some are tolerant species. They reflect the organic enriched and pollution. For example, the Spionidae, you can find it anywhere. That species is specifically for organic enrichment area. And as well as the next organism is mollusk. They respond to sediment contaminant and habitat change. For example, any habitat, any pollutant, particular pollutant pressure on that any habitat, the mollusk will indicate them. Like that, crustaceans will indicate the sediment quality and the habitat structure, while echinoderms indicate stable and healthy marine habitat. How we can using these numbers, how many numbers are there present in the particular environment that home is. We need specific tool to indicate what impact on the habitat. So these are the tools we can use it. These are the tools we can use it to find out the sediment quality or habitat, any changes are there. For example, Shannon Weaver index, using that we can say that benthic diversity on the community complex and the species richness index. In that Shannon diversity itself, there is a species richness index. The number of tags are present at a specific site. For example, if mollusk, they're high abundant. So this particular tool will use it to tell the what impact happens on that area. And AMBI index, this also will tell you the pollution and the tolerance group. And MAIB in integrated diversity and richness. This MAMB, it's more or less similar like AMBI, and it will tell you the diversity, richness, as well as the pollution and the tolerant groups. Finally, the BNTAX access benthic response to the environmental disturbance. There is any impact disturbance happen, how they respond. Like for example, if they tolerate that place or they're very sensitive to that place, this way they respond to the environmental disturbance. If we read only, if you learn about only the organisms, we need to know the how these organisms benefited from the surrounding parameters, like environmental parameters, like temperature, salinity, dissolved oxygen, and the pH, how they helping to build their community. How this temperature or salinity favor to them to get the food or live in their place. So not only the physical parameters, but also the sediment characteristics, the organic matters and the sediment textures. These also important parameters, those organisms which lives in or on the sediments. Heavy metals and the contaminants, like emerging contaminants, those can alter the abundance and species composition. There are anthropogenic, all the anthropogenic, they impact the abundance and species composition, especially heavy metals and contaminants, emerging contaminants. Those can alter the abundance and species composition. Using a specific tool like PCA and CCA, we can say them how the variation happens based on the environmental parameters. Like as you see, I hope you can see the diagram which is there right side. That there is the arrows and all. Those arrows and all physical parameters, and the names which is around there. Those are the organisms, like specific species, we can say. How this specific species are abundant in that particular area based on those parameters. For example, if the organic carbon level is high, the Spionidae particular species abundant will be high. Those thing and all, we cannot manually say. We have to use some tool to officially show them that they are correlated with these parameters. So these tools and all will help you to identify the environmental parameters and their relationship with the particular parameters. And these are the advanced technology which we can use it to improve our analysis. Like how we use manually, we go and collect the sample using grab and identify them under microscope. We are human, we make some errors. In order to avoid the errors, we can use this advanced instrument like ADNA. We can use it ADNA to tell the absence and presence of the diversity. Like that, FTIR, we can use it to composition of the microplastic. And GCMS, we can use it to what are the hydrocarbon presence in your samples. And ICP-MS, we can use it to metals and especially heavy metals. We can quantify the heavy metal, how many heavy, how many are there in our sample. So these are the instrument we can use it to reduce our workload and improve our data. Final conclusion, using benthic organisms, you can tell the coastal ecosystem health, like how they are in particular species, how the, for example, polychaete is abundant, which tells about the organic, and the mollusk. Each groups will explain you about the health of the coastal ecosystem. Like mangrove, there is mangrove in particular organism is associated in that. So that will indicate the mangrove area is healthy. Like that, each each macrobenthic organisms which indicates the health of the coastal ecosystem. So calculate them changes in their diversity, abundance, and community composition will tell you the environmental stress, what type of stress they are having. Using the tools like as I told you to like index to tools, we can strengthen our assessment and pollution monitoring. Which we manually what we can say like this this parameter is directly correlating with this organisms, which we can instead of saying the manually, the software will tell you the correct and exactly what parameter is correlating with that. Combining with the macrobenthas with the eDNA and advanced tooling, using to assess the environment better compared to other methods. Eventually, this all will tell you the health of the environment. Thank you. Thank you, one and all.
Thank you, sir, giving a informative lecture. Sir, one question is there. That is the why doesn't shrimp archery operator prefer the SPF polychaete swarm rather than wily polychaete swarm? Why polychaete swarm? This is the question, sir. Sir, you are
SPS,
audible, sir.
SPS, yeah, yeah. Can you hear me?
Yes. Yes, yes, yes.
Wild polychaetes is sometimes they may carry um, some pathogens like some pathogens they carry because the polychaetes are vector for those pathogens. So that's why mostly they prefer SPS free polychaetes to feed the shrimp. I hope you understand what I said. Hello?
Yes, sir. Yes, sir. Thank you. Thank you, sir, giving a wonderful answer. We are happy to inform you that MSc Marine Biology program for the 2026-2028 batch is now open. This is regarding details our center facility, the characterization service and internship and workshop. This is the open year. Kindly have a look at. Thank you for a wonderful session. Thank you, sir.